Right, if things work properly, you now see the title slide. Is that right?
We see it.
Good. Yeah, so this is a bit of a rough talk because I actually compiled all the work we did on that involves chirality more or less for the first time in one presentation. And I decided to start it off with something which is not directly immediately linked to chirality, because I want to connect back to that at the very end. So try and bear with me. So in contrast to David's work, this is really only an experimental study on addressing the issue of chirality with self-replicating molecules. And we've entered this through our work on trying to make a completely synthetic form of life. So we're not so guided by things like prebiotic relevance. We just want to piece something together. As a chemist, you like to create stuff, and I guess the ultimate synthetic target would be a living something, where the something need not be using the molecule's current life uses. We don't particularly care, although we sneak in some fragments or some components that are used by life as well, but we also don't care about that in particular, as long as it works. And this whole adventure started by chance, by finding self-replicating molecules in our mixtures that we didn't design to be there. They just happened to show up. So let me start there. Let me start with also how they work. So replication can come about if you have two very simple ingredients: a reversible oligomerization reaction and an assembly process. And you see here in this animation those two things happening. You starts off with only doing the reversible oligomerization, and the oligomers here happen to be rings. I don't think it's essential that they're rings. So what you see here is simple building block, only one, that oligomerizes into rings of different size that range from 3 to 6 or 7 or so. And once in such a system an assembly process happens, and that's just about to happen now, it's a slow nucleation followed by a rather faster further assembly. The ring size that does the assembling gets sort of taken out of solution, if you like, and even though it still looks like a homogeneous solution, and replenished by this solution, the stack grows, and the stacking process, the assembly process, thereby effectively drives the synthesis of more of the rings that do the assembly. So this is where you get to self-replication. It's even exponential. If you stir these things, long, thin things will break, and with that, the number of ends will double, and each time, and the growth rate of the ring that makes the stacks is proportional to the number of ends, which we've been able to demonstrate also experimentally. So we have an exponentially growing system where the product inhibition problem that David already referred to is now solved by mechanical energy breaking our stacks into two, or possibly also more and more pieces. So just to recap what you saw in the animation, we start out with something relatively simple, single building block. We can do this with mixtures as well, but I will not touch upon that today. Simple building block, oligomerization reaction that's reversible. Then one of the oligomers happens to self-assemble. The assembly process then drives the synthesis of the ring that makes the assembly in a exponential fashion. So that's where you get self-replication. Now the real mechanism is slightly more complex than what I showed you in the animation, in that material also docks, material that's in solution docks on the side of the fiber, diffuses to the end, and then there it gets converted into the ring size that makes the fiber. So that's how replication works. And this is a building block with which we discovered this whole process. It has files at the end to make the oligomerization possible by oxidizing them to disulfides. Just oxygen from the air in water, neutral pH is good enough. It has a short peptide chain that was designed to form beta sheets by having alternating hydrophobic and hydrophilic amino acids. But it's too short to do this in the building block. It's also too short still to give much assembly potential for the small rings, but by the time you stick 6 of them for this sequence 6 together, there is enough stickiness, there is enough of a tendency now in this hexameric molecule to assemble that you start getting this nucleation, elongation, fragmentation cycle. Now, that's replication. I'll now do a little excursion before touching on the issue of chirality into metabolism and also slightly into compartmentalization, just to show how what we're trying to achieve and how catalysis fits into this. So replication is nice. It's autocatalytic, so the rings that form the stack catalyze effectively the formation of more of them. But for a metabolism, you want additional catalytic activity, as the replicating system should also be able to produce catalyzed reactions that produce molecules that it can use to grow from. So how can a replicator invent catalysis is then the core question. And it turned out it did that already the moment it became a replicator. And the way that works is by converting a building block into this fiber, you basically arrange the amino acids that are appended to it into a beta sheet. So that basically spatially organizes them, and it happens to do so in a way that that generates catalytically active sites and substrate binding sites. For catalysis, it's rather important to have non-protonated lysines, whereas lysines normally, and also in the building block, would be protonated at neutral pH. But if you put a lot of protonated ones, if you were to put a lot of protonated ones together, there would be so much charge repulsion that some of the protons would fly off, and you end up with having non-protonated lysines in the assembly, but not in the building block from which it's made. So that then generates a situation where the assembly, where the fiber has catalytically active sites where the building block didn't. And it also has hydrophobic residues, this building block, which also cluster together to form hydrophobic binding pockets. So this generates a lot of potential for catalysis, and one and the same peptide sequence can catalyze a remarkable number of different reactions. So FMOC cleavage, I mean, if you're not a chemist, don't worry, this doesn't speak to you. The point is, it's a lot. FMOC cleavage, cofactor mediated photoredox catalysis, aldyl reactions, Knoevenagel condensation, hydrogen formation, ester hydrolysis. I'll just briefly mention FMOC cleavage because it connects to compartment formation. FMOC cleavage FMOC is a generally used protecting group for all kinds of molecules, and it's normally cleaved off under basic conditions, but here under neutral conditions, a replicator can do it too because of this non-protonated lysines acting as a base, and probably the protonated lysines helping with leaving group departure of in this case the glycine amino acid that we're using as a model reaction. So we can catalyze this reaction, which in itself is of little interest, but it can be turned into something that is relevant to the system if we can now also catalyze the cleavage of FMOC groups of a molecule that converts it from a precursor state that is not interesting to a sort of more relevant structure that does something useful. And the something useful now is compartmentalization, making a compartment. So here is a molecule that we know from the work of Evan Sprout to be able to produce coacervate droplets. So it self-assembles into a coacervate, which is a liquid-liquid phase separation or a liquid organelle in cell biology. It's a funny phase separated state where there's still a lot of water around the self-assembled phase made of this molecule, and that then forms droplets that exist in a continuous water phase. And they're exciting because they mop up all kinds of stuff that is in solution. It concentrates up hydrophobic things, charged things, and that's an attractive feature if you want to make a compartment and want to make life from it. And the replicator is now able to set this molecule free from a protected form which has FMOC groups that I just introduced previously, but rather special ones that have very polar sulfonate groups attached, so that this molecule is very soluble, does not form a coacervate. the moment you start to cleave these end groups off, you end up with this molecule, which is no longer very water soluble, does form the coacervates, and you see that in this video here appearing, and over the course of about half an hour. And the color here also indicates that where the replicator is, and that's inside. So the replicator now makes compartments and then enters them as we would expect based on this tendency of coacervates to do such things. Okay, so that's just a very brief recap of what we can do with catalysis. And again, I'll try and come back to catalysis at the very end. But let's now switch to the meat of and the central topic of today's session, and that's chirality. And if you look for experimental systems of replicating molecules that address the issue of chirality, it's remarkably little that you'll find. And before we started doing anything, there was only one paper, this one here by Reza Gadiri, that looked not at chiral symmetry breaking, but at enantioselective replication. So it's a replicator based on alpha helical peptide. So there's a template peptide along peptide sequence that folds into alpha helix, and then two shorter alpha helices that are binding to this longer one. And by binding, you basically promote the reaction that ligates the two to make a copy. So that's I think it's very much the same as the replication mechanism that David already explained, except that the copy is now not complementary sequence, but it's identical to the original one. So these are the peptides, not that it's very relevant what the exact sequences are, but the peptides that Gadiri used for this point here is they're long. And in order to get enantioselective replication, you start off with something that is already one enantiomer, where every amino acid, except for the glycine, so a few of them there, will have a chiral center. So you need to have done quite a bit if you take this into an origin of chirality scenario. You need to have done already quite a bit of enantioselection and symmetry breaking before you even get here. But once you get there, it's remarkably selective. So this really only replicates its own chirality shorter fragments into copies of itself. It refuses to act on something that is the opposite chirality. So they did do the synthesis of the non-natural amino acids of those peptides. And it's even able to select for one amino acid mutants, if you like, so where only one of the amino acids has the wrong chirality. That already messes up things. So it's remarkably selective, but you really need already a lot in terms of chiral groundwork to have been done before you get here. Can you do it a little bit more simple? So let me show you now what we did first in terms of chiral selection, and then I'll go to chiral symmetry breaking. But first chiral, so enantioselectivity basically. So we go back to our building block. It's a slight modification from the one I showed you before, but it doesn't really matter so much. This you can make into a hexamer replicator if you just do this in regular buffer, or a pentamer replicator if you do this in the presence of 4 molar guanidinium chloride in the same buffer. I will not dwell on why that is. But there's an important distinction between these two, which I felt when I first saw it was like, where did that come from? Why? And this guy, the hexamer, seems to not care at all about the chirality of the building blocks. And when I say chirality, if we make the all L or all D version of this building block, it will incorporate both quite happily. It's a little bit less efficient at replicating from a racemic mixture than if you were to take one enantiomeric form of the replicator with one enantiomeric form of the food, but it's still fine. So it's completely not enantioselective. Now let's go to the pentamer. That shows rather that's much more effective if you try to grow it from racemic food. It does a little bit, but it's really having a hard time. But if you give it only one enantiomer, then it's much more efficient. So that is enantioselective. Of course, that immediately raises the question: why is there this difference? And this looks like to arise from the different conformations that these different replicators have. So here, and this we knew already from previous molecular dynamics simulations on this work, that replicating molecules can organize the amino acids in different ways. And this is what we've been calling the cartwheel conformation, where basically there's if you like, a certain directionality. So hydrophobic groups on one side, hydrophilic groups on the other side of the beta sheet, and if they point all in the same direction, then this is the cartwheel. But you can also imagine that the hydrophobic phase of one beta sheet wants to pair up with the hydrophobic phase of another, like so. And in that case, you get the pairwise conformation. Now, you would imagine this would be the preferred form if you can do it. And for an even-numbered replicator, where you have even number, then this is a feasible arrangement. But when you have an odd number of building blocks in a ring, then you will have one unpaired strand. And then the alternative of the cartwheel becomes an option, because what would speak in favor of the cartwheel is that it can make, and MD shows this, it can make salt bridge interactions between terminal carboxylates and lysines, but in a funny way. And let me try and explain. So what you get is, when you look at the stack of rings, if you look at the residues that are of two different rings adjacent in the stack, it's a salt bridge between a guy that is one of the residues that is, say, below with of the ammonium of one of the rings below, with the carboxylate above it. And that below above arrangement means you need to point in the right direction in order to do this. So chirality matters. If chirality were opposite, this guy would point in the other direction and forget the salt bridge then. So for this salt bridge arrangement in the cartwheel conformation, chirality matters. For the hydrophobic pairing, hydrophobic interactions are not so direction sensitive. I mean, as long as hydrophobic stuff sticks together, you're fine. This is probably a lot less conformationally dependent and chirality dependent. Hence, the hexamer is fine if it's in this pairwise interaction to just incorporate whatever enantiomer it sees. Whereas when you're in a cartwheel, like the pentamer probably has to be, then you really do care about the chirality of your building blocks. That's our current thinking. I think in view of the time, I'm going to skip this because it basically reiterates what I already said. Now with this system here, if you do an experiment where you take racemic building block, you seed it with a one-to-one mixture of an all-L replicator and an all-D and another all-D replicator, then they both grow, and they grow by incorporating the correct enantiomer with about 90% fidelity. So this shows that basically what Gadiri had already shown before, that you can be selective. It now also shows it with a somewhat shorter peptide, where we now have 4 chiral centers in the building block. And we've shortened the peptide a little bit further, and I think even with 2 chiral centers in the building block, maybe even 1. I need to reread our own paper. It's still the principle still holds. So that's nice. It's chiral selectivity, but probably not the core still of what our session today is about, and that is about enantiomer selectivity. Now, most of our previous work, we're started off with a chiral building block already, and then of course the chiral symmetry breaking has already been done by us choosing the building block. Would be much more interesting if we can move from chiral to non-chiral building blocks, because then there is no symmetry to start with, and we could stand the chance of breaking it. And this we found by chance when we saw, well, not the non-chiral building block, but the chiral symmetry breaking. When we moved from peptides, where most of your amino acid, nearly all, will have a chiral center, to nucleobases, which don't have any chirality to them per se. Certainly not if you connect it in this way to the same diethyl core. So if you take this building block and you allow it to oxidize in water, roughly neutral pH, Here's what happens: the building block disappears with time, and you get selective production of a trimer, this molecule here, which, like I showed you before, assembles into fibers. Now the building block has no chirality to it. The conformations that the trimer can adopt, if we home in on this system here with 3 aromatic rings connected by disulfides, because of the 90-degree torsional angle that disulfides want to have, the conformations that you can adopt are chiral inherently. Of course, racemic probably to start with, but conformationally, this one is a mirror image of that. And in solution, these probably interchange very rapidly. But in a fiber, where these guys are stacked on top of one another, this interchange of conformation will be much harder because it probably needs to occur in synchrony with all the neighbors. And that means it really is it's a much more difficult proposition. But you can still, in principle, racemize if you want. All right, so now what we found is if you take this building block, convert it to trimer, that at T0, when you're still at the building block stage, and you put it in the circular, you measure the ECD, the circular dichroism of it, you would not expect to find a signal, and you don't. But after 4 days, when it's mostly trimer, you do find a CD signal. And if you repeat the experiment 10 times, in this particular case, 5 times it's a positive, and 5 times it's a negative CD signal. So it looks like we get chiral symmetry breaking in every experiment. And it's completely random as to which of the 2 enantiomeric arrangements we make. Now, I think I should stress the chirality here is not a molecular chirality, it's a supramolecular chirality. It's chirality that comes about probably by freezing in a chiral conformation through an assembly process, or by an arrangement within the assembly that is chiral and slow to interconvert. So the different and you can even get different types of CD spectra. So there appear to be 2 types that both exist in positive and negative chirality. This we call type 1, this we call type 2. We have no idea what gives rise to them. It probably has something to do with the supramolecular organization, or it may have something to do with the conformation of the ring within the supramolecular organization, or maybe both, but it's very hard to decipher. But this organization is heritable. You can do a seeding experiment with either one or the other enantiomer of type 1 or one or the other enantiomer of type 2, and you retain, at least in the first few seedings, you retain that organization. Here's the data. So type 1 seed in both chiralities gives rise to, if you've added to a system that has not yet developed any CD spectrum, then it gives rise to the same CD signature. Type 2 seed the same. There's a gradual, if you keep doing this, there's a gradual conversion, I think, from type 2 to type 1 that seems to be preferred, but you tend to keep the same chirality. So chirality is inherited, organization also, but somewhat less robustly. Then the question is, how does this happen? And we considered two possible mechanisms. The most simple, if not trivial one, is just nucleation determined. So your very first assembly can only have one chirality. It's either D or L plus or minus. And if that is the mother of everything that happens next, then all the offspring will have, as I just showed you, will have that chirality. So that's the nucleation. Primary nucleation governs. So primary nucleation is a rare event, and then secondary nucleation occurs faster than making another primary nucleus, and then whatever happens to be a chirality of the primary nucleus basically dictates the chirality of everything that follows. But that's pretty trivial. A somewhat more elaborate or complex mechanism is known from the from people that study crystals and the chirality of them. It's called Vietnam ripening. And this is a case where you can, if you start with achiral monomers and they can racemize in solution, or you can start, sorry, you can either have achiral monomers, you can also have chiral monomers that are able to racemize. Both scenarios work, but simplest is achiral monomers. If they can crystallize in crystal packings that are chiral, then they can do this, and again, initially stochastically, you have either the S or the R form. If you keep stirring a system like this, then eventually you will de-racemize, and this seems to be robust. So this is known in the crystal field. Any system which crystallizes a conglomerate, that means you have separate RNS crystals. If you stir them, they will spontaneously de-racemize, and you'll get to only, after a while, you only have one chirality left. So, and this means that for, I think about 20 or so % of all organic molecules, or roughly thereabouts, crystallize as conglomerates. So this is a mechanism that is able to de-racemize, provided you have a conversion in the say solution phase or an achiral monomer, to de-racemize and break the chiral symmetry. So this is so the question then is, what happens in our case? Are we dealing with this nucleation dominated symmetry breaking, or are we dealing with a Vietnam ripening like process, which would be rather unique because it's known for 3D crystals, but not for these 1D assemblies that we're making. So if you then carefully monitor whether, one way of addressing this is carefully monitor whether the circular dichroism signal, so the signature, the extent of symmetry breaking correlates with the formation of the trimer. Where the trimer formation, we would argue, would correlate with the trimer assembly, because normally you would also get tetramer in systems like this if there's no assembly, or at least not assembly in these fibrous forms. And you look carefully, then so you see here how the trimer grows, and you see here how the chiral signal grows, and you see it lags behind. So even though not a big effect, the fact that it lags behind suggests we may also have at least a contribution of the Vietnam ripening in our chiral symmetry breaking system. This is done at a high concentration, relatively high concentration of monomer of 1 millimolar. If we go lower in concentration, then our nucleation becomes harder. So you now see, sorry, I should have mentioned, this is the different lines here are different repeats of the same experiment. Here again, different lines, different repeats. But now you see there's only 2 cases where the trimer emerged. The other ones just failed to nucleate. And the 2 cases here, you see the chirality emerging. And now it seems to also maybe, well, more or less keep, it's hard now to say whether it keeps track or is lagging behind. And it's an even more pronounced issue with nucleation when you go even lower in concentration. So this, at least the first at high concentration experiment suggests maybe Vietnam ripening occurs. Then we did an experiment where we made a racemic mixture from fibers that we mixed the L and the D fiber together in as much as possible 1-to-1 ratio. And then, so this was, the fibers were already quite old, and then we stirred it and monitored over time whether actually the racemic mixture would de-racemize, which is what you would expect for Vietnam ripening determines chiral symmetry breaking, but you wouldn't expect it if it's all governed by primary nucleation. And indeed, you see the de-racemization. Apparently, we're not very good at mixing 1-to-1 because there's a bias. These are 2 repeats of the same experiment, a bias towards one of the 2 enantiomers in this experiment. So it looks like we can sort of, that Vietnam ripening is indeed occurring here, and that it's possible to de-racemize to get to chiral symmetry breaking at a supramolecular level with this particular monomer that has this nucleobase in it that I showed you. I will not show you the data, but it seems that that is a reasonably general feature, because about half the molecules we've made with which you can make replicators where the monomers have no chiral center also show the same behavior. So it does look like with the reversibility of the assembly and the fiber and the self-assembly, and there's the reversibility of the ring formation and the self-assembly combined, chiral symmetry breaking can be a fairly general feature. So now, why did I start my talk off with catalysis? Well, what I showed you is that you can make assemblies, replicators catalyze reactions. I showed you that replication can also break symmetry, but only at a supramolecular level. And being a chemist, I really like molecular level chiral symmetry breaking. So what we're now working on is see if we can use these supramolecular chiral fibers to do catalysis of a reaction that then should produce molecular level chirality by making that process enantioselective selective through the environment that the supramolecular chiral fiber provides. So that's we haven't got there yet, but I'm optimistic that we will be able to manage that. I should wrap up here. I think in view of the more focused nature of this talk, I'll skip my usual final slide saying that simple ingredients like small molecule building blocks and oligomerization plus self-assembly gives you a lot. But it includes chiral selection and chiral symmetry breaking. And I should thank the people who did the work. It's particularly single out Mark Geert here, who was really instrumental. He did all the work on the chiral symmetry breaking, and Yannick Geiger, who's now started this independent group in Strasbourg, who did the enantioselective selective replication work together with Xiao Yang, who's not on this picture. He's there, I think, on that picture, who had the intuition that the pentamer should behave differently than the hexamer. I did not believe him at all, but I'm glad I let him let him be and do it, because he was right. I still don't gather how he can possibly have predicted this, but he was right. Thanks for listening, and I'm very happy to start a discussion.
Well, thank you so much, Sijbren.